So let's get started. I have the pleasure of introducing our speaker this week, Dr. Fred Korley. Before I talk about Fred's work, remember we were talking about, you know, if you have real complex problems in medicine and you want to have a comprehensive solution to them, it's really essential to use advanced engineering and computational techniques to essentially come up with a solution that actually works. And traumatic brain injuries, TBI, has been one of these problems. And as Fred will describe later on, there's been so many solutions, medications, models, and things like that. They all fail. And that requires a new generation of researchers who are equipped not only with the medical knowledge of the field, but also advanced engineering and computational methods to create these solutions. And we have Fred for that. So a few years ago, Department of Emergency Medicine that I have the pleasure of working with was trying to form a sort of core faculty who can focus on TBI, traumatic brain injuries, from different points of views and come up with new solutions for that. And Fred, who was at the time at Hopkins, was running one of the largest and the most comprehensive studies called HeadSmart that he will talk about that later on, I'm sure, during the presentation, that looked at molecular biomarkers that might be sort of informative and add substantial information to the models that we form from TBI. So Fred has been one of the leading researchers in a couple of larger scale studies that are happening across the nation. One of them is HeadSmart that we will talk about. He's also one of the active members and leaders of something called Track TBI that I'm not sure if we talk about that, probably point to that. So we have basically the pleasure of having a researcher who's as knowledgeable as any engineer that I've seen when it comes to technology for molecular assays and analyzing them using computational technique, as well as being an emergency medicine physician who's focusing on TBI. And it's a fantastic, basically, opportunity for us to learn about that. Before I let Fred start, I have to add that Fred has been instrumental in recruiting students to our program and co-mentoring students in Department of Computational Medicine and Bioinformatics, including Haley. And that's been a fantastic opportunity for our department. Without any further delays, I'll leave the floor to Fred, and we're happy to have you there. Oh, you don't need that.
And I'll turn it off. And thank you so much, Kevin, for that excellent, wonderful introduction. Some of which were, slightly embellished, but that's how most introductions go. The part that I take credit for, though, is the awesome Haley. And if you don't hear anything else, I think that Haley is super awesome, and she's been instrumental in a lot of what we're going to talk about. And while I wait for some more people to come in, first I want to talk about the emergency department. This is what it looks like. Has any one of you been to the emergency department before? Yeah, it looks like that. It looks like that. We see patients everywhere, in the waiting room, in the hallways, but never in the bathroom. We don't do that. And that's really important because most of the decisions that we make are time sensitive. And part of the reason I show this picture also is that we're usually faced with information overload. So it's not unusual that I am taking care of, a cardiac arrest patient, and then a nurse comes in and sticks an EKG in front of me for a chest pain patient, because those EKGs have to be reviewed within 5 minutes of being obtained. And so, I'm running a cardiac arrest. Someone is sticking an EKG in front of me, and then, I may get a call almost at the same time about, a transfer patient who is going to come in, or there may be another patient next door who is critically ill. And so, we typically, have a ton of information overload. And with every passing year, there are new diagnostic tests. And so, there's a lot of information coming from all those different sources as well. So being able to drill down knowledge and information from all those varied data sources is critically important. And I think that that's where you all fit in and I'm hoping that as I talk, you begin to sort of have ideas about how you can help us be more successful in taking care of traumatic brain injury patients. So I want to tell you about how we take care of traumatic brain injury patients. And first of all, I'm going to say that there's a huge burden of traumatic brain injury evaluations in the emergency department. But our current evaluation is inefficient, and it's also overly optimistic. So every year in the United States, we see almost 5 million ED visits for traumatic brain injury, 5 million. And out of that 5 million, about 2.5 million are actually diagnosed with traumatic brain injury. And these are only patients who come to the emergency department for traumatic brain injury. There are tons of people with traumatic brain injury who never seek medical care. The majority of patients who come in with traumatic brain injury or suspected traumatic brain injury get a CAT scan. Now, the CAT scan is horrible. It's a lot of money, yes, and it doesn't tell you a whole lot. So what the CAT scan does is it tells you if you're bleeding in the brain, period. Sometimes, you can see some signs of axonal injury, but the brain is made up of a whole lot of cells that are not blood cells. And so if you damage, neurons and axons and astrocytes, those usually don't show up on the CAT scan. And although we get CAT scans on 80% of this 4.5 or almost 5 million patients, only 10% of those CAT scans produce information that we can use. Only 10% of those CAT scans are positive. So the majority of CAT scans are actually negative and uninformative. So you're going to see these 4 letters, GOSE. The GOSE is a scale we use for measuring clinical outcomes in traumatic brain injury. And it was developed in Glasgow. And 1 is equivalent to death, and 8 is that you're back to your functional baseline. And so you're going to see this a whole lot, and I just want to orient you in terms of what the GOSE is. And we have a number of different ways for categorizing the GOSE. One of the usual cutoffs is 4 or less. And for a GOSE of 4 means that you're at home and you can exist by yourself at home, but you can't get out of the house by yourself. If you need to go shopping or go out of the house, you're going to need help. And typically, if someone after TBI is not able to go out and shop independently and do activities of daily living independently, then we say they have a bad outcome. Because we're hoping that we can get people to the point where even if they need help with someone around, they can at least go out and be independent. So we did a study called the Head Smart study, where we examined patients who came to the emergency department with traumatic brain injury, and about half of the cohort had a GOSE of less than 8 at 3 months. Less than 8 means you're not back to your baseline mental status, and that you probably are still having headaches, you're having difficulty concentration, difficulty with sleep, and so on. And when the patients came to the emergency department, I asked the ER physician, this person in front of you, what are the chances that they are going to still have symptoms at 3 months? How accurate do you think the ER physicians were? Any guesses? So, someone comes in with head injury, and then we ask them on the day of injury, do you think the person you're seeing now is going to keep on having symptoms at 3 months? Not very accurate. Yeah, 20%. So our emergency physicians were very optimistic, and they pretty much thought that everybody was going to be fine. And they said only 4% of patients were not going to be fine. And part of the issue is that we currently classify traumatic brain injury as mild, moderate, or severe. So bad. Tell someone who used to be able to go to work but because they are not comatose, we're calling them mild. But they can't go to work or they can't go to school because they are having headaches and they are having difficulty concentrating. And I remember this one patient who said she was a public school of public health student at Hopkins, and she said, I'll go into the bathroom, I'll shower, shampoo my hair, and then forget that I shampooed my hair and do it all over again. Or talk to someone on the phone and don't realize that I just talked to that person. But we call those people mild, which that's a huge disservice, to those people. So essentially, my point is, we do a really bad job of diagnosing traumatic brain injury. Now, and when it comes to clinical trials for traumatic brain injury, here's what we do. So, although we have one term, traumatic brain injury, there are different subtypes of traumatic brain injury. You can have a contusion, you can have hematoma, you can have cerebral swelling, you can have diffuse axonal injury. There are a number of different subtypes of traumatic brain injury. Here's what we do when we do clinical trials. We put everyone in the same basket. And so, these are, some studies that have been done in traumatic brain injury, and all of them have failed. All these studies have been negative. People come up with a new drug, they test it, and it shows that it doesn't work in traumatic brain injury. Why is that? It's because we're acting like we're treating cancer. Can you imagine doing a clinical trial where you say, I have a drug for cancer, and so I'm going to treat everyone the same? I'll treat breast cancer and prostate cancer and lung cancer all the same. That's what we're doing to traumatic brain injury. And no wonder the treatments are not working. It's because we're lumping everyone in the same basket and treating them all the same. So it's not about equality, it's more about equity. That also in these clinical trials, we treat everyone equally, which should not be the case. There are some patients who are going to need more of the dose of a certain drug, right? You may have someone who has a more severe injury, and so they may need 20 milligrams of a particular drug, as opposed to someone else, who has less severe injury or smaller body size, who needs a smaller dose of that drug. But we currently, treat everyone the same. We give the same doses to patients. And we do not have a good way for monitoring how well patients are responding to the various treatments we give them. So we're essentially driving blind. And typically I show a video about driving blind, but that's essentially what we do. We, without paying attention to the different subtypes of patients, we lump them all in the same basket. We give them the same treatment. We have no way of monitoring the treatments to titrate treatment according to the effect of individual patients. And then when the trials fail, we question why this is happening. So this is my conceptual framework. This is sort of how I view the world of traumatic brain injury. That initially there is the primary brain injury. We can't do anything about that. Primary brain injury is where you've damaged some brain cells, and sort of is the mechanical impact. And the only way that can be prevented will be through public health campaigns and wear helmets and wear seat belts and so on. So I can't do a whole lot about primary brain injury. Then there are the pre-injury factors that I can't do a whole lot about either. We know that the more educated you are, the better your outcome is going to be if you have traumatic brain injury. So stay in school, keep on studying. We also do know that people with premorbid psychiatric problems tend to have a harder time with recovering from traumatic brain injury. And so these are premorbid conditions that we can't do anything about. Here's what I can do something about: secondary brain injury. So after the primary brain injury, there is secondary brain injury. And the secondary brain injury is caused by a number of different things. One is cerebral hypoxia, which essentially means low oxygen levels in the brain. So, the brain is a highly metabolic organ that uses a lot of energy. And if your oxygen levels are low in the brain, you're not going to be able to perform the functions that are necessary. The next important thing is decreased cerebral perfusion, which means you have low blood flow going to the brain. And these are the two main areas where we try to intervene. Some of the causes of cerebral hypoxia or decreased oxygen level in the brain are things like poor oxygenation. So the traumatic brain injury patient may also have lung injury, and so they're not getting enough oxygen. Poor ventilation, which speaks more to the carbon dioxide levels, that they're not breathing well and exchanging gases well. And if you have a high CO2 level, that can cause constriction of the blood vessels, and so parts of the brain will not get enough blood flow. There's also low hemoglobin. If trauma patients can lose a lot of blood, and so the low hemoglobin can also cause decreased amount of oxygen in the brain. And then you can also have increased metabolic rate, and that sometimes can happen when you have a seizure. Some traumatic brain injury patients have seizures, and some of them also have infections, and all that stuff can increase the metabolic demand and lead to cerebral hypoxia. There is also decreased blood flow to the brain, which is decreased cerebral perfusion. That can happen from systemic hypotension. So the brain injury patient has a liver injury as well, and is bleeding in their belly, so their blood pressure is low, and so they're not getting enough blood to the brain, and that's causing additional brain injury on top of the primary brain injury they already had. And then this other one is also really important: increased intracranial pressure. So whenever you have injury to the brain usually swells, and the pressure in the brain goes up. And if the pressure in the brain is up, you need higher blood pressure to be able to get more blood into the brain. And so essentially the equation for cerebral perfusion pressure is your systemic arterial pressure minus your intracranial pressure. And the higher the intracranial pressure goes, the lower your cerebral perfusion pressure is. And so these are the areas that we can intervene on. These are the things that we can do to prevent secondary brain injury. But how do we measure secondary brain injury? We have no tools. There's really just no good way to measure secondary brain injury. There's some new diagnostics that I will talk about, that allow you to measure things like brain tissue oxygen, and that allow you to measure cerebral perfusion pressure. But all of those have limitations. My hypothesis is that it's all in the blood. That we have lots of things that we can measure in the blood. We can measure proteins, we can measure metabolites, we can measure RNA, and by looking at these analytes in blood and looking at how they change over time, we will be able to better monitor secondary brain injury. And so if you don't hear anything else in this lecture, that's the one thing I want you to remember, that my main premise is that it is in the blood, that by measuring analytes in the blood and by measuring how those analytes change over time, we can get a good sense of secondary brain injury. So I'm not just making this up, that we already have evidence that there are some proteins that you can measure in blood that can give you a sense of what's happening in the brain. So this year, we had 2 blood tests that were approved by the FDA for traumatic brain injury. They're GFAP and ECHL1. So GFAP, glial fibrillary acidic protein, is a structural protein found in astrocytes, and essentially is released anytime you have damage to brain cells. And ECHL1 is an enzyme that's found in neuronal cell bodies. And again, anytime the cells die, they release this enzyme. And that's what we've been measuring. And so a study was done just this year called the ALERT TBI trial that looked at 1,780 patients. And I'm going to walk you through this table. And so they, there were 107 of them had a positive CT scan. I told you that majority of the CT scans are usually negative. So in this group, 1,673 patients had negative CT scan. And a negative CT scan, though, doesn't mean they didn't have TBI. It just means they were not bleeding in the brain. Now, this test is really good in that out of the 107 people who had a positive CT, the test was able to pick 104 and missed 3. And those 3 that were missed, were actually one of them, even though they had a positive CT scan, when they did an MRI, the MRI was negative. And the other 2 had tiny, tiny, tiny amounts of bleeding in the brain, which, they didn't do anything about anyway. So, and, the negative predictive value is actually also pretty good. And so this 668 people are people who did not have a positive CT scan, and they also had a negative test using this blood test. So these are actually CT scans that can be avoided, the 668 people. So first point, that GFAP and ECHL1 are good blood tests for identifying, those who are going to have a positive CT scan. And then using, data from one of the studies I've been working on called the TRACK TBI cohort, and I won't bother you with all the details, but essentially TRACK TBI is a multi-center study that enrolls TBI patients across the country and follows them for the clinical outcome. And based on this one blood test, GFAP, and looking at the data, we can categorize the GFAP values into 3 zones. There is a zone of people with super low levels, and these are people who do not need a CT scan, and they're not going to need surgery. So essentially, even though they may have TBI, their levels are so low that you don't even have to worry about getting a CT scan in these people. And then there are the middle zone people who need a CT scan, but they're probably not going to need surgery. And so if, for the military, for example, these people in both the green and the red and the yellow zones are people who do not necessarily need to be evacuated from the field because it's really unlikely they're going to need surgery. And then there are people in this red zone who are people with the highest GFAP levels. And then those are people who absolutely need a CT scan and most likely are going to need some sort of surgical intervention. So, the data is unfolding. I mean, we're not using these tests clinically yet, but we're slowly building the database. And the takeaway is that these blood tests are really good, A, for identifying those who have bleeds in the brain, and then also B, for risk stratifying people, trying to figure out who is super sick versus who is not that sick. So this is another study that I'm hoping to submit for publication either tonight or tomorrow, where we're looking at 900, I'm sorry. Yeah. Crudely, yes. Yes, because, and I guess with the next slide, I'll show you that. So the mild, moderate, and severe is based on the GCS. So GCS is the Glasgow Coma scale, and it's essentially, so yeah, it is related, but the blood tests do a little bit better than GCS, which is what the next slide is going to talk about. So we looked at 946 patients, and we looked at 5 different proteins here. Okay, so the proteins are GFAP, CHL1. There are a couple more proteins we looked at: S100B, NSE, and high sensitivity CRP. So again, what did I just do? Okay. All right, it's back. Okay. Just to get everyone. Oh, it's gone again. It's back. Okay. All right. So, for each protein, we divided the values into quintiles, and the y-axis is, you remember the GOSE, and 4 or less is that you can't operate independently outside your home. So these are the number of people who didn't do well, essentially. And as you can see, for most of these proteins, those who were in the highest quintile, they did not do well at all compared to those in the lowest quintile. Um, and especially the first three, this, and that, you can really distinguish between those who did well versus those who did not do well. These two didn't work out so well. Now, how many of you are familiar with the area under the receive operator curve? Awesome. Okay. So GCS, like I said, is what we use now. It's based on whether the patient is talking, if their eyes are open, and if they are moving their extremities. So you don't have to do any tests, any blood tests. You don't have to pay any money. This is based on a clinical exam, and how well the clinical exam does right now. The AUC is 0.623. When we added these two proteins to the AUC, to the biomarker, or to the GCS, the AUC went up to 0.853. And typically, an AUC of 0.9 or greater is like excellent. 0.7, 0.8, it's good, and less than that, it's probably not clinically useful. So this is good. This is good, and not too many biomarkers are able to do this. Yes. What is the gold standard? So that's the GOSE. That's the scale where we ask people how well they're doing and see if they're able to operate functionally at home or not. The scale of 1 to 8. Say it again. Yeah. So this was measured actually 6 months afterwards. So essentially, these are people who didn't do well at 6 months after injury, and these blood tests were obtained on the day of injury. It's pretty powerful that on the day you come in, I can tell you how well you're going to do in 6 months. So we also, and this is sort of where I'm going to need your help a whole lot, is that we also look to see whether we can follow these biomarkers over time, and whether these markers can tell us how well a patient is doing and how well they are responding to treatment. And so we worked with Dr. Alam's lab. Dr. Alam is the chief of trauma surgery, and he has some pigs that are TBI models. So they essentially hit the pigs in the head, they give them TBI, and then they monitor them over time. And they decided to test a drug called valproic acid. And so this is the group that received the test drug, valproic acid. These are the people who just received normal saline, which is essentially just water. And so we measured the levels of GFAP, which is the protein I've been talking about, over time. The time scale was from 0 hours to 10 days. Who can tell me the difference in the two graphs for VPA versus normal saline? Any takers? Anyone? Shout it out. What's the difference in GFAP levels between those who got water only versus those who got the experimental drug? Come on. Yes. 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 Essentially, that the levels in the biomarkers, in this particular biomarker, was a lot lower in those who got VPA compared to those who got normal saline. Now, if I tell you that this biomarker is measuring cell death, because this biomarker is a structural protein that sits inside the astrocytes, and is only released when astrocytes die. So if some animals are having lower levels of this marker, what does that suggest? Anyone? Less cells are dying, yes.
We published a paper on it,
Yeah. Yeah. So. Yeah. So this is actually an astrocytic marker, and yep, and then the lesion size also went down. So the blood is actually showing exactly the same thing, and so we think that we can actually monitor how well people are doing by looking in their blood and telling how much brain cell death is going on. We looked at a second protein called neurofilament light chain, and this is an axonal protein. And we saw the same thing, that the levels of this marker were lower in people receiving, the animals receiving VPA compared to those not receiving VPA. And essentially, this is sort of where we're going with this line of work, that not only are we interested in measuring on the day of injury what's happening, but we can follow patients over time and get a sense of if they're doing well or not. Right now, when we do clinical trials, what happens is that we have to wait till 6 months to be able to tell if a drug is working or not. But with this, we should be able to tell in a shorter time period whether your drug is actually causing less brain cell death or not. And but it's not just these proteins. Metabolites can tell us something as well. Not just metabolites, but RNAs can tell us prognostic information as well. And the challenge with monitoring over time is that you start having a whole lot of data. And remember the first picture I showed you about the emergency physician who is taking care of a ton of different patients and has this information overload? It is going to be really hard to decipher what to do with all this continuous data biomarkers. And so we need novel techniques to be able to make sense out of these longitudinal data, and especially to see how the different proteins change in relationship to each other. Remember the slide where I showed you 5 different proteins and 3 of them were actually associated with the outcome. There are a number of different proteins that can be looked at, but we have to understand how they're changing individually and how they're changing with respect to each other. And that's sort of the challenge we have, and that's something that we could use your help with solving. So that's what Alam is doing as the next phase, and so they've started a phase 2 trial with VPA. And for my part, I'm not as interested in VPA. I am more interested in being able to monitor how well patients are responding, regardless of the treatment they're receiving. Yeah. Yeah. Which he's actually also tested. He's looking at some HDAC specific inhibitors as well. And yeah, no, it's awesome. I want to tell you about some of our ongoing projects, where we're going to have a ton of data and we'll know what to do with. One of them is Boost 3 trial. So Boost 3 just got funded by the NINDS, and Boost 3 essentially stands for Brain Oxygen Optimization in Severe TBI. So what's happening in Boost 3 is that we're going to enroll a little over 1,000 patients, and patients are going to be randomized to either ICP monitoring only. What I mean by that is they put a probe in their brain to measure the pressure. Remember I told you perfusion was important? So they put a brain in to measure the pressure, and they direct treatment based on pressure monitoring. So you're not going blind, you're going based off of what you can measure. And then some people are going to get both ICP monitoring and partial brain tissue oxygen monitoring. Now, this is what partial brain tissue oxygen monitoring looks like. So they drill a hole through the skull and put a probe in a normal part of the brain. And that probe measures the oxygen level at about a 2 centimeter radius and gives you a value. And usually you don't want anything less than 20. So the treatment, will be targeted towards getting that level higher than 20. So some patients are going to get only ICP monitoring. Some patients are going to get ICP and brain tissue oxygen monitoring. And then we're going to see which patients are going to do better. Thank you. Yes, yes. Which is why blood tests are going to be better. And that's my whole point with that. That what that probe does is they put it in normal brain tissue. So you have traumatic brain injury, you have one area of the brain that is like all messed up, for lack of a better scientific word, and then you have the good part of your brain. And the probe is going to be placed in the good part of the brain, and the probe is going to measure a small radius around the tip of the probe. And it does so happen that, if you've had severe traumatic brain injury, there's also spasming of blood vessels. And so even the good areas can still have decreased blood flow. But you can't tell me that for sure, if the normal brain areas oxygen is fine, it doesn't really tell me that the injured brain area is fine as well. And this is why I think that the biomarkers are going to do even better than this tool. Because with biomarkers, as I've shown you already, I can tell you how many brain cells are actually dying. And I can sort of measure, the global amount of brain cell death. So we're in the process of submitting an application called BioBoost, where for patients enrolled in this funded trial, we're going to get blood samples, and we're going to get longitudinal sampling. We'll do every 8 hours on the first 2 days, and then twice a day for the next 7 days, and then continue getting samples up to 6 months out. And so we're going to have a ton of data. And because we have blood, we should be able to measure all kinds of proteins and other analytes in these. And yes, we'll be doing all of that as well. Yeah. So the boost is going to use exception from informed consent. The parent study is going to use exception from informed consent, where it's a whole process where you do community consultations, and you tell. Yes, yes. So this is, but this particular study is exception from informed consent. Yeah. Yeah. I would say though that for BioBoost, which is the biomarker portion of the study, we are going to get consent, and we're going to get consent from the legally authorized representatives. So we will. Our current plan is that we'll draw the initial sample. We will hold it. We'll look for a family member. If we can find a family member, then we will do the additional sampling. If we can't find a family member, then we will discard the initial sample. We just don't want to miss that initial sample, so we're going to get it regardless. And Haley knows we've done this in other studies. Yeah, this is all severe TBI. This is all severe TBI. All severe. All GCS 3 to 8. So yes, they can consent, and yeah. Yeah. It's all severe TBI patients, and yes, there's a whole process for doing exception from informed consent studies. Matter of fact, we had gone to the DoD asking for funding for BioBoost. They didn't fund us because they don't fund exception from informed consent studies. Yeah, so yeah. I know I'm okay. There's another study that I'm working on, where we're actively enrolling patients now. It's called the Hyperbaric Oxygen in Brain Injury Treatment Trial, where essentially we put people in a chamber um, um, with compressed oxygen. As I told you earlier on, hypoxia, can be one of the causes of secondary brain injury. And so the hypothesis here is that if we give you a ton of oxygen under pressure, we're going to be able to get a whole lot more oxygen to the diseased brain cells, and hopefully people can get better. So people are randomized to different doses of this oxygen versus standard of care where you just get regular oxygen, and we're following people to see who's going to get better. And we're looking for funds right now to actually collect blood samples in these patients as well, so we can be able to test, whether this treatment is actually causing a decrease in brain cell death based on blood samples. The last study that I want to talk about is a study that Haley is intimately familiar with, and she's enrolled all 20 subjects that we have in the study right now. And we call it Optima TBI Pilot. One of the things you're going to notice is that for any clinical trial to be successful, you have to have a cool name. So if any of you is interested in doing clinical trials, you got to work on the mnemonics. In this study, what we do is we randomize more of the mild TBI patients. Again, I hate that word mild, but these are the non-comatose TBI patients. We give them 6 grams of fish oil per day for 1 month, and then 1.2 grams of fish oil per day for 2 additional months. And you get randomized to either that or to placebo, which is olive oil for 3 months. And the pills look pretty similar, I guess, apart from if you break them or if you burp after taking them, and you smell the fish oil. We follow these people we get blood samples when they come in, and then at 2 weeks, at 1 month, and at 3 months. And we're going to look to see if the levels of these biomarkers of brain injury change over time. So we have 20 patients now, and we're hoping to add a few more before we start analyzing the samples. That's an exclusion. Yeah, it's an exclusion criteria. Yeah, you can tell them how many times you've run to the ED, and we got excited, and then patient was on fish oil, so we couldn't enroll them. Yeah, the challenge is that yeah, you need a lot of people who've been on that to be able to answer that question. So I think, yeah, that would be a good question to answer down the line. One of the things that we're also doing is that we're collecting red blood cells, and so everyone has like a DHA intake, right? And so we're gonna get the red blood cell DHA intake, and we can know exactly sort of what the baseline is, and see whether those with low baseline do better, than those who are already saturated. So great question. And so we're working with some engineers to come up with a way where we can monitor the levels of these blood biomarkers in essentially a continuous fashion, and then hopefully beam the results to like an iPhone or to a monitor. But again, these are going to pose analytical challenges because the more frequently you measure things and the more values you have, the greater the need for advanced analytics that would be able to give clinicians some sort of decision support and some sort of binary decision making. And there are a ton of people who've helped to make this possible, and I thank them all. And I'll pause for questions.
Seeing that GFAP was astrostatic in nature, and yes, I understand that blood samples are easier to draw, but are you seeing any of these biomarkers in CSF?
Absolutely, yeah, and for all the studies, yes, we are collecting CSF as well, and a lot of severe TBI patients, have drains in the brain, so it makes it easier to collect CSF in them. But so-called milder patients, usually don't have any of those drains, and so it limits the applicability, in the mild patients, but for sure in the severe patients. And I do think that at least the initial data suggests that the CSF values are more accurate than blood, which is to be expected. Yes.
Have you looked at all at variation in where the injury occurs within the brain and whether that affects the GFAP levels in the bloodstream?
Yes, that is something that we're planning to do, but haven't done yet. So Track TBI enrolled a total of 2,700 TBI patients. We've analyzed samples in the first 1375. To be able to effectively do subgroup analyses like that, you need more N. And I actually question whether we're going to be able to even do that analysis with 2,700 patients, because the challenge is a lot of people have a lot of different things going on, right? You don't just have one area of the brain damage. You usually have 3 or 4 areas of the brain damage. So again, this is the opportunity for you guys to help out. Kevan has an algorithm for being able to automatically quantify lesion size and lesion location. And again, as all these studies enroll patients, the NIH and other funding agencies are requiring that for all of these major trials that are going on, all of the data needs to be put in a particular warehouse. And about a year or 2 after the end of the study, everyone can have access to it. So, as you plot your career going forward, these are going to be databases that are available to you. The images, the blood test results, and the clinical data, they're all going to be available to you.
So just to follow up on that, the reason I was asking about that is recently found that there is a population of astrocytes in the brain that's located in 2 different regions that actually express GFAP under normal circumstances. So it might be really interesting to try to see if maybe the reason why this biomarker shows up in some cases is because the astrocytes on the outside of the brain are
So
damaged.
they express GFAP in normal circumstances.
Yeah, so we can chat afterwards.
Yeah.
But basically, we found that there's a thin layer of astrocytes expressing GFAP on the outside of the
On the
cortex.
outside, because
Yeah,
they all have it in the inside.
Right.
But they may have it on the outside as well. Yeah, so maybe those with injuries to that area may have even higher levels than those without injury to that area. Is that the
Exactly.
hypothesis maybe? Yeah. Yeah.
So when you injure the brain, the astrocytes kind of come in and infiltrate and create that protective layer. That's probably what you're talking about. That'd be a gold one.
Yeah.
Yeah, good thinking, Josh. Yeah.
Any other questions? If not, I would like to thank our speaker once again. It was wonderful, and we enjoyed your talk.
Thank you. Thank you.